Good evening, this is Mr. Wintermelon, and this is the worst video you can watch to learn about cell structure and function. This is a series of videos, and we will start with how we can recognize cell structure in a typical O-level paper. So what are cells? Cell is the most basic unit of life. And can you see them? Yes, you can see them, but not so much with your naked eyes. We need the help of something known as a microscope, which you will learn later. So to give you a scale of how small cells are, you can actually open a browser. Boom! Now that you have opened your browser, you can go and Google scale of the universe. Okay, the first link, scale of the universe, click on it. Then you will get a flash app. So after I click the start button, I will get some music. I assume it's copyright, so I'm going to mute the audio until I manage to mute the music. Okay, I have managed to mute the music. So this little app down here is actually quite good because I can scroll in and out and give people a sense of how big, like I said, human is compared to an egg. But of course, this is actually quite a small thing. I can't really zoom it. So what we can do is this. You can go to the page info. Then you go to media. There's an object down here. It's a SWF file. So you can actually save it. So you will get a flash file. So once you manage to download the flash file, you can click on it. Then you can boom, full screen. Where is full screen? Full screen. Ta-da! Okay, then you can use your mouse as well to scroll around. So this is much better than looking at the website. Okay, now, so the size of human is down here. And if we scroll down, we are talking about cell level because this chapter is on cell, how small cells are. So this is an ant. Imagine how small an ant is. This is the first cell. Bacteria, they are made out of cell. This is the largest bacteria. And the rest are a lot smaller. So we can kind of see it with our eyes, but it's not something that we do it every day. A grain of salt. This is the smallest thing we can see with our naked eyes. But of course, not all of us, our eyesight is that good. And human egg, something that comes out of non-menopause ladies after their puberty, every cycle. Something that come out from their body, unless they are pregnant, of course. So we can actually see them, just that maybe it's too bloody. That's why we don't notice. Now, human skin cell, you can see, is smaller than the smallest thing that we can view with our naked eyes. So it's much smaller. Then white blood cell, red blood cell, you might have heard of it. Then chromosomes. And as we go smaller, okay, you can see viruses. So what we are talking about, the scale of the thing that we are talking about is around this size, and this size, compared to human, which is here. That's why we need special instrument for us to look at or observe and to study things that are so small. Not dismiss. Okay, red blood cell. Of course, we can go in a bit further, smaller. So after viruses, we go to DNA. These are what we call molecular level. Just now is cellular level, bigger, and even smaller is molecular level, DNA. This one is not in your syllabus, but I think I will talk about it. It's phospholipids bilayer. This one is one of it. It's called phospholipids. This one is 2 of it. Then if you go smaller, you can see water molecules. Then I think the smallest I will go. Hydrogen and helium is so small. Hydrogen is part of the water molecules, which is important. This one is a glucose molecules. So these are the things that we are going to discuss in this series of video. So now let us go back to the actual lesson. So first of all, please make sure you have downloaded the syllabus document. And if you are unaware of where to get this, I have actually created a video. The link to the video can be found in the description or the information below. So have this PDF downloaded from SEAB. Then also make sure you have a A4 size notebook to take down the notes in this video, because I believe writing is important for you to internalize what is being taught. So if you open the PDF, you will see that in this chapter, what you have to know is identify cell structures, including organelles. So you might ask, oh, this is new to me, what is organelle? So organelles is a specialized subunit within a cell that has a specific function. So imagine in an organism, we have our organ. In cell, we have organelle. So in an organism, we have brain, which the specific function is for us to think and feel. Then we also have our specific organ, stomach, which specialize in storage of food as well as digestion of protein. In cell, they have organelle instead of organ, because they are much smaller. Another example of organ is our hand, which is very useful apart from the month of November. So we will learn about how to identify structure as well as organelle. So as you have learned earlier, cells are very small. How do we observe and study them? By microscope. So earlier, we can see that you have to learn 2 types of microscope, the light microscope as well as electron microscope. So first, let's talk about the light microscope. Light microscope is named light microscope because it uses light. Light will reflect from the specimen, then we will see the light and we will see the specimen. Something that come out from light microscope will look like this. So you can see that it's colored, and we can see that you can't really make out what is it, but I can tell you later what is it. Then we also have our electron microscope. So electron microscope, of course, it uses electron, and how electrograph, things we see from an electron microscope, looks like this. So you can straight away tell the difference. Electron microscope, right? The magnification is much higher, while the light microscope, the magnification is smaller. But there's no color in electron microscope. Why? Because it doesn't use light. It uses electron, reflection of electron. And the things that we can see is much smaller, and you can probably visualize or see that, oh, it looks something like sperm. Well, it is. So the light microscope, the specimen is actually from testicle. So electron microscope, there is the sperm where they are being generated. So for electron microscope, most of the time not most of the time, all the time the specimen is dead. Because if they are not dead, we cannot cast or stitch them onto an electron microscope. For light microscope, the specimen can be live. So that's the difference between a light microscope and electron microscope. Light microscope, magnification is lower. Electron microscope, the magnification is higher. Light microscope, it can be colorful and live. Electron microscope, only gray, but you can Photoshop color onto it, and the specimen is dead. So that's the difference. So if you refer back to the syllabus, you will have to know which organelle you can observe in light microscope and which organelle or structure you can see under an electron microscope. So what I need you to do now is to sketch a typical animal cell and a typical plant cell in your notebook so that you can refer to it and label the organelle and structure. You may refer to your textbook for a not so good reference. But like what I said, it's a not so good reference. So why would you want to refer to it? It is probably better for you to refer to the typical cell that I'm going to show you. So typical O-level diagram, the cell will look like this. So the one on the left is the animal cell and the one on the right is the plant cell. So pause the video, sketch it in your notebook. So I assume that you have finished sketching, so let's move on. First, let's talk about how what you can observe under a light microscope. So under a light microscope, what you can see of an animal cell is this, and what you can see of a plant cell is the one on the right. So first of all, cell membrane. Label them. The nucleus is the round black color thing that's in the cell, very prominent. Then you also have the liquid called cytoplasm. For plant cell, you have chloroplast. Also, you will see a vacuole for plant cell. Then, of course, plant cell, you have cell wall. So these are the structures as well as organelle that you can observe using a light microscope. Pause the video, copy them down. When you switch to an electron microscope, what you can observe is a lot more. Under an electron microscope, you can see organelle known as mitochondria, ribosome, rough endoplasmic reticulum, or we can short form them as RER. So you might notice some black color dot on the rough endoplasmic reticulum. So those black color dot, right, actually they are ribosome, which we have labeled earlier. Other organelles will include smooth endoplasmic reticulum, or we call them SER, as well as Golgi apparatus. So these two organelles is actually quite problematic for us to recognize because they look almost identical, or it's very difficult to tell them apart. So what I noticed is for Golgi bodies, right, they are actually not linked together. So for smooth endoplasmic reticulum, we can see there's some bridge joining one stack to another. So for O-level, for Cambridge, they represent smooth ER using those joining bridge, while for Golgi bodies, it's completely separated. Each layer or each stack of membrane is completely separated. So these are the organelles you have to know for your pure syllabus. For combined syllabus, I will probably make another video for it. You have to know a lot lesser than this. Lastly, I would like to take this time to point out, please take note of how we label in biology. If you look at my labeling, I use straight line, not arrow, straight line, and the line touches the organelle that or the structure that I'm referring to. So this will apply throughout, and this is especially important for your practical. So when you are labeling, please take note, you do not use arrow. Just use a straight line and make sure your straight line touches what you are referring to instead of air molecules. So we have come to the end of this lesson, and in our next lesson, we'll learn about the function of organelles.